Okay, I think we're good to go.
All right.
So thank you for the time. What was your name?
My name is Gene Smith, unless wrong.
Okay, got it. And that's not your real name, is it?
Correct.
You are utilizing this name because people know you in the less strong ecosystem and stuff, correct?
Correct.
Lovely. And we're here to talk about some of the most interesting research I found at Vitalia, which was a company that you're building alongside the post that made you quite blow up, like famous on Less Wrong, in my mind, right? And what exactly are these topics that you're describing and discussing?
So the main topic of the post was adult intelligence enhancement via gene editing. This is something that I long thought was impossible before I embarked on this 5-month research project to see, okay, if this, can this be done? And also, if not, why is it impossible? I guess to give you some background here, I've been interested in intelligence enhancement for a while because there's so many positive, both societal and personal externalities that go along with higher intelligence. In many ways, intelligence is sort of the defining human, characteristic, and I think the key to a lot of our success as a species. So I've been interested in intelligence enhancement for a while, and genetic augmentation seemed like quite a promising way to do this, simply because there's so much variance in the human genome that you could potentially alter to affect intelligence. Um, for a long time, I assumed that the only way to, make genetic alterations would be to the germline. Yeah, embryo selection or perhaps embryo editing someday. Um, but I was somewhat concerned about the progress of AI and started to think that perhaps even if we were able to make the technology to enhance embryos, we wouldn't necessarily have time for those children born via the technology to grow up. So I started examining faster ways that might work perhaps within 5 years as opposed to within 20. And a friend of mine was investigating a kind of a separate topic of like how difficult it would be to do gene editing in embryos, and realized that the the editing technology itself has gotten so good that you might not even need to do editing in embryos. In other words, you could make a significant number of edits in adult cells and expect that to not necessarily cause a large number of off-target edits or insertion and deletion errors. Um, I immediately replied saying, I think this is impossible, and here's why. Um, but it seemed promising enough that we decided to take a deeper look, which led to this, collaboration in this research post.
Fantastic. And so just to clarify, you looked into augmenting intelligence for human adults as defensive tactic towards the rise of AI, if I understand this correctly, but not just, right?
Yes, that was part of our motivation, and not really as a defensive tactic, against AI. I think it's unlikely that in the long run humans are going to be competitive with digital intelligence. It's more as a key technology to bridge this period during which we're creating artificial general intelligence. I see, human intelligence enhancement as a sort of key tool in um, to overcome that technical challenge.
Okay, but all this sounds very science fictiony.
Yes, I
And
recognize.
you talked about gene editing, and we've talked about this before, but can you walk us through a little bit how would that work theoretically or in
Sure.
practice, even if you've done enough experiments on this?
Sure. So it is fairly complicated, but the basic idea is you want to edit a significant number of genes in a significant portion of the cells in the brain. How exactly you do that can be fairly complicated. Um, so challenge number one is like, how do you actually know which genes to edit? And there we actually have we have quite a bit of help from recent genome-wide association studies. So to give you some context here, genome-wide association studies are these giant, multi-100,000 person studies that gather data about what people's genes, what genes people have, and also their whether or not they have diabetes or heart attack, or what their IQ score was, or how tall they are. And if you gather enough data, if you gather enough of these genotype phenotype pairs, you can basically figure out which genes are actually causing someone to be taller or smarter, or have a lower risk of disease. So we have reasonably okay intelligence predictors today from genome-wide association studies, but that's still an area for significant improvement. Yeah.
Fantastic. And the most of the gene editing that's happening is happening with a small subset of genes, right? But what you're trying to do is multi-gene.
Right. Correct.
Okay. And there's not many people doing this type of research or actually work specifically.
So there's definitely research on multiplex gene editing. There's no one really focusing on this in like a therapeutic context. There's some very limited examples of this in like CAR T cell therapy that are just now starting to kind of pick up. But there's no one really focusing on this in the context of polygenic disease risk, for example, or let alone intelligence.
Fantastic. So, okay, and we understand the scope of your work now, right? Tell us a little bit about the story behind how you go to be building this business now.
Oh, I guess two more things I should mention in terms of like how we actually get this done. Once you know which genes to edit from those genome-wide association studies, the next two steps are actually making an editor that works and can target the genes that you want to modify. And then perhaps most challenging of all, getting those editors to the correct cells. I think that last issue is actually the biggest barrier to making this work. Getting a sufficient number of editors into a sufficient percentage of the cells in the brain, in my view, is probably the biggest bottleneck to making this happen right now.
Okay. And how would you go about doing this? How would you go through the blood-brain barrier?
Yeah. So again, to get editors to all the cells in the brain, the most convenient way to do this is to basically inject something into the bloodstream and to have it basically diffuse into the brain. But there's a couple challenges. As you mentioned, one is the blood-brain barrier, which is basically this fairly impermeable set of cells that line the blood vessels that kind of perfuse, diffuse throughout the brain. So you have to design some kind of carrier for the editing proteins, or the editing, the DNA for the editing proteins. And that carrier molecule has to be able to, that carrier, sphere has to be able to get through the blood-brain barrier. And once it's through, it has to diffuse, and then get taken up by cells. And that's quite challenging. There's a couple of promising ways to do this. One is these things called exosomes, which are basically like little bubbles of fat. And you can attach you can basically attach little things called peptides, which are like small proteins, to the outside. And those can help the exosomes get past the blood-brain barrier and also get taken up by neurons and glial cells in the brain.
Okay, fantastic. This is a tested method already, right? From what I heard, you've already done such work with bubbles of fat that have editors in them.
Yeah, so the ones that are probably tested a lot better than exosomes are called lipid nanoparticles. Lipid nanoparticles were used in like the COVID vaccines, for example. They're quite easy and cheap to manufacture. The issue with those is that they mostly end up getting taken up by liver cells, which is great, actually, if you want to target a polygenic disease in the liver. But it's not so great if you want to go to the brain.
I remember that you were saying that looking at the liver and how the liver itself can distribute some of these editors was an interesting research subsector for you. Is that something I understood correctly?
Yeah, that's correct. So, the company that I formed in the aftermath of this, this post success has it's mostly focusing on the liver as a first target organ for a gene, a multiplex gene therapy.
Okay, fantastic. So before I jump on the business side of things, because I think there's interesting sides of the story there as well, I wanted to ask you, You presented to me a timeline of what you want to do and how to effectively tackle this. And I would love for you to describe this to us again, if it's possible for you. You are looking at the liver first. You're trying to solve something very difficult afterwards, and then you want to do intelligent augmentation. Can you tell us a bit more about what you're trying to solve first and why?
Sure. So I won't get too much into the details, but basically, there's delivery to certain organs is easier than others. The liver is probably the easiest organ of all to target, apart from maybe like the muscle cells. But there's many different diseases that have that involve multiple risk genes. And some of those diseases are in fact like liver-based conditions, such as like alcoholic fatty liver disease, or a couple others. So one of the most sensible targets to go after is it's one of these liver diseases.
Got it. And then the second target is
Um, the second target, second target organ or second target disease.
the second target disease.
So I mean, there's other types of polygenic liver disease we could go after.
I remember that you were telling me that you're trying to tackle Alzheimer's first before intelligent augmentation.
Right.
And I wanted to learn a bit more about why you think that is easier than intelligent augmentation.
Yeah.
If you think it is easier, if it's the same methods that you're using, and if it is a realistic treatment that you believe can enter the market over the coming 5-10 years.
Sure. So, I mentioned that like liver it's the first organ we want to target because it's probably the easiest to go after, and there are polygenic diseases in the liver. But if we can kind of like reasonably solve delivery of editors to brain cells, then we could start going after brain-based diseases. You might wonder like why Alzheimer's first and not intelligence augmentation first. The basic answer is that intelligence augmentation requires you to edit a significant number of genes. And also, when this therapy is first tested, there's going to be some amount of risk. We'll do what we can to decrease risk with animal testing, with testing in organoids and cell cultures. But any new medical therapy you roll out, especially one that has kind of a whole new modality of treatment like this one, there's going to be some risk involved. So I think it's going to be it's a much more straightforward sell if you are trying to use the therapy to cure some disease that's definitely going to kill someone in a couple of years if you can't cure it or at least treat it. There's other reasons to do this as well. There's fewer genes. You basically don't need to edit as many genes to have a significant impact on Alzheimer's as you do to have a significant impact on intelligence.
Got it. So less genes, more urgent for the patient.
Right.
Arguably faster for you to trial this in states like Montana, which I'm saying that they allow for people to utilize these not approved fully medical procedures if they are terminally ill. That's something that we've learned. But okay, so we talked a bit about the business. We talked about your end goal. There is a whole technology that you'd like to explore as one of the first people doing this. But you're not just the first on the technology and the targets. You're also a first in how you actually raise money or trying to raise money with this business, right? So you started with a blog post on Less Wrong.
Yes.
And that got a couple of hundred upvotes and likes and a lot more comments, right?
Yeah.
And Less Wrong is a blog that people here know about, but not many people outside of here know about. What happened next? You posted this, which we're going to link.
What happened next? So, after 5 months of work with my collaborator, we posted the results of our research, and the post just exploded. I think within 48 hours, we had 2 emails from venture capitalists asking if we were planning to start a company. And at the time, we were looking for a grant, but there was so much interest from investors that we decided, maybe we can just start this as a company right now. And so we did.
And I know some of these names. Do you would like to mention them or not really?
I'd prefer to avoid that for now.
Got it. Okay, that's fine. and but they were pretty interesting big people in the space. So how much money do you want to raise right now,
Yeah.
for how much equity, and then also how much you feel like you will need for you to actually put this gene editing technique to intelligent augmentation implementation?
That's a good question. So right now we're raising our seed round. We're raising 1.5 million at a $7 million cap. In terms of how much we see this costing to get to, let's just like start with a kind of closer goal of like how much would it take to get like a working therapy into use. And there we're not entirely sure, but my estimate is maybe 20 to 50 million, depending on the sort of costs of clinical trials for humans and also where we conduct those. If we did US FDA trials, I think it would take significantly more. My guess is at least a few hundred million.
Got it. And go ahead, please.
Just to expand upon that, I think that's probably the cost for one successful treatment. For let's say intelligence enhancement, there's significantly more barriers to that one. We need a better understanding of the genes involved, and we also need to solve delivery. Delivery is a partially solved problem right now, but not to the degree that you would want to have a significant impact. So exactly how much that's going to take is not entirely clear to me, but my guess is like at least a few hundred million.
Got it. And why are you in Vitalia and Prospera? Do you find value being here? Do you think doing something in a special economic zone like Prospera is going to help diminish the cost and the time to market?
So I think the most immediate answer to that is that one of our investors invited us to come to Vitalia. But I think since I've arrived, I've realized kind of the wisdom of that invitation. I think Vitalia has been more open and eager to hear ideas like intelligence enhancement via gene editing than I think any other group of people I've met. It's really actually kind of stunning just how open people are to this, this idea that to many seems, quite out there. It's difficult to get when you tell people that like you can edit the genes in their brain and make them smarter, it sounds like such a science fiction idea that like very few people will actually take you seriously. And if they give you the time, you can walk them through the science and the approach and like why you think it will work. But your base sort of hit rate of being able to get into a deeper conversation with people in the broader world, it's quite low. Most people just are not even interested in hearing about the idea. In Vitalia, it's been much different. People are much more open. They are actually very interested in this idea when I explain it. So it's been a great place for us.
Well, I'm happy I met you here, that's for sure. From this point onwards, the questions are going to be a bit more social, right?
Sure.
Have you had backlash from presenting this idea? And if so, what was the backlash?
Surprisingly, we haven't really had backlash so far. Granted, we haven't really presented the idea that much. We gave one presentation in Vitalia, and that's basically it. All of the rest of our writing has been online, and there we have received backlash. I think I made a funny compilation of reactions to our post when it first went up. People on Less Wrong were kind of very, very open to and excited about the idea. But there's kind of, entire communities on the internet whose like sole sort of shared value is like hating on ideas like this. So they were obviously less receptive to the idea. Yeah, I mean, I could get into details if you want.
No, I think that's fine. Any last remark I'd like to give to the mic about the technology, the targets, the goals? I mean, really, why is this motivating you? Why do you find this so interesting for you? Are you trying to become even more intelligent? Are you trying to make yourself 7 feet tall? If you can do that, I don't know. I'm just trying to get your feedback on why this is important for you.
Yeah, that's a good question. I think that when I think about a future I want to live in, it's quite easy for me to envision a positive future with genetic augmentation. Um, and the positive impact we could have with this technology is really, it's hard to overstate how big of an impact this could have. I mean, you could potentially, if you can solve delivery, you could potentially cure every major disease. I'm not even joking about that. Like, you could potentially cure like everything. Um, you wouldn't necessarily be able to like completely end aging itself, but you could have a gigantic impact on people's quality and quantity of life. So I think that in itself is motivating enough. But I think what really excites me is the prospect of going beyond sort of my current limits. Not just for me personally. Really, what excites me the most is like large groups of people going beyond their current limits. Um, I mean, imagine for a moment that like you could comprehend calculus as Newton did, or relativity as Einstein did, or art like Picasso did. These are all human experiences that we might one day be able to have. In fact, we might be able to go even beyond that. So I think to me that's like a vision of the future that makes me feel positive and makes me feel like this is worth working on.
And it can be achieved.
And it can be achieved. It's going to be hard, but I think there's a clear route for us to like make a workable therapy that makes money. And once we have that, then we can invest in the R&D necessary to do intelligence enhancement and cure Alzheimer's and stuff like that.
Got it. Well, I think that's it. Gene Smith, thank you very much.
Thank you.
Good luck.